This lesson is on speciation. Here are the objectives. Now, speciation means how new species form, how they evolve. So let's review our definition of a species. A species is a group of organisms that can interbreed to produce viable, fertile offspring. Viable meaning that they survive. And this definition is only one definition of species. There are many, and it's called the biological species definition. Key here is that if a group of organisms can only interbreed with each other to produce viable fertile offspring and not with any other organisms, then this leads to the concept of reproductive isolation. This population of organisms is isolated reproductively from any other population and can't swap alleles with any other population. So imagine this is a gene pool containing all of the alleles at all of the loci in a population. So I'll put some alleles in here to indicate that. And here's another population with its own gene pool. Now, I'll call this one 1 and this one 2. If the organisms in population 2 can only interbreed with each other to produce viable fertile offspring and not with population 1, then there's no way for alleles to move from one population to another. The populations are reproductively isolated. And that means that if a mutation arises in this population, let's call that allele E, there's no way for this allele to move over into this population. They're reproductively isolated, and so now they can start to diverge genetically and phenotypically. And we would call these two populations two different species. Again, the biological species definition is focused on reproductive isolation. It's a group of organisms that can interbreed with each other to produce viable fertile offspring, but not with other populations. Note that there are problems applying this in a lot of cases. You can't apply this to species that reproduce asexually, for example, bacteria. You also can't apply it to species in the fossil record, because it's based on reproduction, and there you're just looking at fossils. In cases like these, we might use other species definitions, like, for example, the morphological or structural species definition, where you look at the physical traits of an organism and use that to classify it. And there are numerous other species definitions as well, but the biological species definition is an important one. Let's focus a bit more on this concept of reproductive isolation because it's so central to the process of how species arise. Reproductive isolation refers to barriers that prevent interbreeding or the production of offspring that can survive and reproduce. Again, this is like our definition, our biological species concept. The idea here is that gene pools stay isolated from each other so that they can continue to diverge over time. And note that when we're talking about barriers here, we're talking about biological barriers, not just geographic barriers. It can include geographic barriers as well. So how can you have biological barriers to reproduction? Well, you could have barriers to mating or fertilization. These are sometimes called prezygotic barriers because they prevent the formation of the zygote in the first place. So prezygotic barriers. So for example, things like having different habitats or different mating times, different mating seasons, different courtship behaviors. These kinds of things can keep populations from mating with each other even if the species are relatively similar. Related species of birds, for example, may have different courtship behaviors and therefore not mate with other species besides their own. There can also be barriers to survival or reproduction of the offspring after the zygote is formed. So these are called postzygotic. An example here is that the offspring may be non-viable. They may not survive because of genetic or chromosomal incompatibility. Alternatively, the offspring could survive but be sterile. An example here is a mule formed from the mating of a donkey and a horse. The mule survives, but because it's sterile, genes from or alleles from the donkey population are not getting into the horse population, and vice versa. They both end up in the mule, but don't go any further. And so reproductive isolation of the donkey and horse gene pools remains intact. Let's look a little further then at how speciation happens. We'll look at two forms of speciation, allopatric and sympatric speciation. We'll start with allopatric. The word allopatric means different land. Allo means different, and patric means land. So This is speciation happening in different lands. What that means is we start off with geographic isolation. That's what allopatric means. So geographic isolation stops gene flow at first, and then later biological barriers to gene flow develop as the populations evolve. So let's say you had a single population of a species of mice, and a river changed course one day and separated that population into two populations. They're still the same species, just in two populations. But now they're geographically isolated, and so any mutations that happen in this population will not be transferred into this one down here, and vice versa. And so you can imagine that over time, they will start to accumulate mutations, and if the environment on the two sides of the river is different and so provides different selective pressure, then we can start to evolve differences in the two populations, and they can start to be genetically and phenotypically different. So now what you've got as a result of mutation and then natural selection is genetically and phenotypically different mice. Again, this could happen because the change in the course of the river geographically isolated them and stopped gene flow between the two populations. Now, if the river were to change course again and disappear, it's possible that when they came back together, they would still be able to interbreed, and so now the gene pools would mix again. And if that were still possible, we would just say that they are still one species. But it's also possible that if enough change has occurred, then now there may be not just geographic reproductive isolation, but in fact biological barriers to reproductive isolation as well. For example, as a result of different foraging behaviors on the two sides of the river, they may have started to mate in different seasons. And so now when they came back together, they would be mating at different times and their gene pools would never mix. If that were the case, now they could actually overlap in the same geographic distribution, and because of these biological barriers to reproduction, their gene pools would never mix. And so now they would be two different species. Again, they could coexist, but their gene pools would still be isolated. So that's allopatric speciation, where initially geographic isolation stops gene flow, and then later, even if that geographic isolation is removed, gene flow still does not occur because of biological barriers. Next, we'll look at sympatric speciation. So sim here means same, and pater again means land. So this is speciation happening in the same land. In other words, there is no geographic isolation. Species are developing in the same location. So if that's the case, how is gene flow interrupted? Well, an important example is polyploidy, and this mostly happens in plants, and in fact is very important for many of our crop plants. So polyploidy refers to having multiple full sets of chromosomes. What's happening here is an error in cell division in meiosis is leading to gametes that have more than the usual number of sets of chromosomes. So errors in cell division are giving us more sets of chromosomes than we would expect. For example, in meiosis, imagine that these are two chromosomes of a homologous pair, and instead of separating in meiosis 1, two of them go into one of the daughter cells and none go into the other. So if this were a 2N organism, this is still going to be a 2N gamete. And so instead of having a 1N gamete, now we've got an extra set of chromosomes. If the gamete that were formed from this ended up being used in fertilization, we would have an extra set of chromosomes. And so the offspring's ploidy, its number of sets of chromosomes, is going to be different from that of its parent and also that of its siblings. And that is going to reproductively isolate that new polyploid plant, because if you don't have a matching number of sets of chromosomes, you're not going to be able to reproduce together. You're going to have mismatches in the way the chromosomes line up. So you need to find another polyploid. So this is why sympatric speciation by polyploidy is most common in plants, because polyploid plants can reproduce with other polyploids, or they can, importantly, self-fertilize. The easiest way to find another polyploid plant to reproduce with is to reproduce with yourself. Or plants can also often reproduce asexually, meaning they don't have to find a polyploid partner at all. So that's why sympatric speciation by polyploidy is most significant in plants. So that's an introduction to speciation, including the important concept of reproductive isolation and a look at allopatric and sympatric speciation.